Okay friends, now let us talk about isozymes. Now what are isozymes actually? Isozymes are enzymes with slightly different subunit contents. Okay, so as you can see, there are for example, we take an enzyme which is mammalian lactate dehydrogenase or LDH, which exists as 5 different isozymes in mammalian body. And why they are differing in all this at all, because these enzymes need to function in many different places. Sometimes they need to function in liver, sometimes they need to function in muscle cells, then white cells, then brain cells, then red blood cells, kidney and heart. So there are different cells, different types of cells, because the cell of liver is totally different than the cell of brain, cell of our red blood cell is different than our cell of our muscles. So they are destined to do different types of work, they're destined to do different specialized purposes, and they are placed in different organization of cell. So that's why for their activity that lactate dehydrogenase to show in different cells, it is varying from one place to another place. For example, in heart and in his liver, this lactate dehydrogenase need to produce something, some different kind of enzymatic subunit arrangement, because in heart they need to do different types of work rather than in brain. So when it presents in brain, it need to do different work, different sets of, or the activity, or the amount of activity it needs to show is varying, not actually different type of work, because an enzyme is destined to do a particular job too, but still this lactate dehydrogenase, so actually there are different tissues express different isozymes forms, because of it actually favors that cell lining to work properly. That's the basic part actually, I think. Okay, so as we can see in this case, this lactate dehydrogenase is actually showing 5 different forms. So it actually based on 2 different types of units. One is the A, another one is the B. So if we arrange 4 A subunits, we make a homotetramer, and this can be found in liver. So you can find this A4 in liver. Now we also find the homotetramer made up with only B unit, B type of unit, and we can find it in heart. So both of them functions on their own manner. Okay, now we can find different types of mixed arrangement, like 3A 1B, we can have it in muscle, we can have 2A 2B, we can have it in white cells, brains and all these cases. We can have 1A 3B, we can also find in kidney. So there are different parts are like that, and not only one each type of say lactate dehydrogenase are found, but also the mixture of all these things. So if we look at the liver, then we can find mostly A4 type of isozyme, and we have few A3 B type of isozymes in liver. That's the case. If we look at this kidney, then you can have mixture of B4, B3 as well as A2 B2. Okay, if we look at brain, we have much more A2 B2 and AB3. If we look at white blood cells, we do not have any kind of species like A4 A3B or something like that. We only have A2 and B2. So it's a homodimer arrangement in this case, as we can see. So 2A and 2B arranges themselves in case of the white blood cells. So it's actually depending upon the type of function they need to do, depending upon the efficiency they need to function inside the cell, it actually varies. Okay, by regulating the relative amounts of A and B subunits, they synthesize different types of isozymes. The cell of various tissue control which isozyme forms are likely to assemble, and thus which kinetic parameters they predict. That's a very important point. So the kinetic parameter will also change depending upon the change of isozyme. So other things are remains the same, because the enzyme functions as it generally functions, but still what varies is the kinetic parameters which are incorporated with the enzyme, because as we are varying the subunits, we are varying the different kinetic parameters. The effectivity, the affinity with the substrate, and the productivity, and all these things will be varied, and it is depending on the demand it is actually needed. Okay, for example, in muscle cells we need to do it differently, in heart cell we need to do it little bit more differently, in brain we need to do some differently, so in different manner. So that's why it's actually varying. So that's the basic part, and this is an example of lactate dehydrogenase that we have seen for having different structure relation of this subunit, and is this subunit arrangement is tightly controlled by the cell and type of cell we are in. Okay, so that's it, and I hope that's gonna help you. Thank you.